Hello friends! In today's video, we're continuing with Food Microbiology, Module 1, Part 3. We'll be zooming into the tiny world of light microscopes. From simple bright field to glowing fluorescence and the cool contrast tricks of dark field and phase contrast, each one shows us a whole new hidden universe. Let's dive in. Microscope and its optical principles. A microscope is an optical instrument designed to produce enlarged images of small specimens that cannot be clearly visualized by the unaided human eye. Microscopes work on the physical principle of magnification, where the image of an object is magnified so that it can be visible. The substances that can only be seen with a microscope are called microscopic substances. Microscopy is further divided into three branches: optical microscopy, electron microscopy, and X-ray microscopy. Microscopy, importantly optical microscopy, began with the discovery of the first microscope by Antony von Leeuwenhoek. In an optical microscope, the rays of light are passed through a series of glass lenses to produce a magnified image on the observer's eyes. Compound microscopes are the most common type of microscope, mostly used for research and teaching purposes. In the case of an electron and X-ray microscope, an electron beam is created which produced a digital magnified image of an object. The magnification of these microscopes depends on the type of lens used in the system, which produces images of different magnitude and resolution so that they can be viewed. Microscopy is important in different areas of science like histology, cytology, and bacteriology. Refraction. Refraction is the bending of light as it passes from one medium to another, such as from air to glass or from water to air. This bending occurs because light travels at different speeds in different substances. When a light ray enters a medium where its speed changes, its direction also changes, causing the ray to bend at the boundary between the two media. Microscope lenses operate on the same principle of refraction. These lenses are typically convex lenses, which have a curved surface that bends incoming light rays inward. When light passes through a convex lens, the rays are refracted and directed toward a common point. Refractive index. The refractive index, n, is a numerical value that describes how much a material slows down the speed of light. It is calculated as n equals speed of light in vacuum divided by speed of light in the medium. A higher refractive index means light travels more slowly through that material and bends more when entering it. For example, the refractive index of air is about 1.0, while glass has an index around 1.5. The difference in refractive index between two materials determines how strongly light will bend at the interface. In microscopy, a higher refractive index improves the amount of light entering the lens, which enhances image clarity and resolution. This is why oil immersion lenses give better results. Focal point. The point at which all refracted light rays meet after passing through the lens is called the focal point, or f-point. This is the place where the image becomes sharply focused. Focal length. The focal length is the distance between the center of the lens and the focal point. Lenses with a short focal length bend light more strongly and provide higher magnification. In contrast, lenses with a longer focal length bend light less and produce lower magnification. This relationship between focal length and magnification is a key factor in the functioning of microscopes. Numerical aperture. Numerical aperture, NA, is an important optical property of a microscope objective lens. Numerical aperture is defined as the product of the refractive index of the medium between the specimen and the objective lens, and the sine of half the angle of light entering the lens. It is expressed by the formula Na equals n sine theta, where n equals refractive index of medium, air, water, or oil. Theta equals half of the angular aperture, the angle at which light enters the lens. Light optical microscope. A light microscope is a commonly used optical instrument that uses visible light and a system of lenses to magnify small objects that cannot be seen clearly with the naked eye. It consists of an illumination source, a condenser to focus light on the specimen, objective lenses for magnification, and an eyepiece for viewing the enlarged image. The basic working principle of a light microscope is the refraction, bending, of light. When light passes through the specimen and lenses, it bends and forms a magnified image of the object. Microscopic magnification varies greatly depending on the types and number of lenses that make up the microscope. Depending on the number of lenses, there are two types of microscopes. Simple light microscope, it has low magnification because it uses a single lens. And the compound light microscope, it has a higher magnification compared to the simple microscope because it uses at least two sets of lenses, an objective lens and an eyepiece. The lenses are aligned in that they can be able to bend light for efficient magnification of the image. The modern types of light microscopes include 1. Bright field light microscope, 2. Phase contrast light microscope, 3. Dark field light microscope, 4. Fluorescence light microscope. 1. Bright field light microscope. This is the most basic optical microscope used in microbiology laboratories, which produces a dark image against a bright background. Made up of two lenses, it is widely used to view plant and animal cell organelles, including some parasites such as paramecium, after staining with basic stains. It is composed of transillumination light source, commonly a halogen lamp in the microscope stand. A halogen lamp, also known as a tungsten halogen lamp or quartz iodine lamp, is an incandescent lamp that has a small amount of a halogen, such as iodine or bromine, added. The combination of the halogen gas and the tungsten filament produces a halogen cycle chemical reaction, which redeposits evaporated tungsten back onto the filament, increasing its life and maintaining the clarity of the envelope. Two lenses, which include the objective lens and the eyepiece or ocular lens. An eyepiece, also called an ocular lens, is the lens that you look through in a microscope or other optical device. The objective lens first collects light and forms an image. The eyepiece is placed near this image and magnifies it so it can be seen clearly. The amount of magnification depends on the focal length of the eyepiece. The nosepiece has about 3 to 5 objective lenses with different magnifying power. It can move round to any position depending on the objective lens to focus on the image. Objective lens is made up of 6 or more glasses, which make the image clear from the object. The typically ranges are 4 times, 10 times, 40 times, and 100 times. The condenser is mounted below the stage, which focuses a beam of light onto the specimen. It can be fixed or movable to adjust the quality of light, but this entirely depends on the microscope. They are held together by a sturdy metallic curved back used as an arm and a stand at the bottom, known as the base of the microscope. The arm and the base hold all the parts of the microscope. The stage where the specimen is placed, allowing movement of the specimen around for better viewing with the flexible knobs, and it is where the light is focused on. 2 focusing knobs, the fine adjustment knob and the coarse adjustment knob, found on the microscope's arm, which can move the stage or the nosepiece to focus on the image. An aperture diaphragm, also known as the contrast, which controls the diameter of the beam of light that passes through the condenser. In that, when the condenser is almost closed, the light comes through to the center of the condenser, creating high contrast. But when the condenser is widely open, the image is very bright with very low contrast. Working principle. The working principle of a bright field microscope is based on passing light through a specimen to create a visible image. Light from the source is focused onto the specimen by the condenser lens. As the light passes through the specimen, some parts absorb or scatter light differently, creating contrast. This transmitted light then enters the objective lens, which provides the main magnification and forms an enlarged image. In some microscopes, a projector lens flips the image so it appears upright. Otherwise, the image appears reversed. The enlarged image then passes through the eyepiece lens, which further magnifies it before it reaches the viewer's eye. Because specimens are rarely in focus immediately, the user adjusts the distance between the lens system and the slide using coarse and fine focus controls to obtain a sharp image. Advantages. The name bright field is derived from the fact that the specimen is dark and contrasted by the surrounding bright viewing field. Simple light microscopes are sometimes referred to as bright field microscopes. Bright field microscopy is very simple to use, with fewer adjustments needed to be made to view specimens. Some specimens can be viewed without staining, and the optics used in the bright field specimen. It is adaptable with new technology, and optional pieces of equipment can be implemented with bright field illumination to give versatility in the tasks it can perform. Disadvantages: by using an aperture diaphragm for contrast, past a certain point, greater contrast adds distortion. However, employing an iris diaphragm will help compensate for this problem. Bright field microscopy can't be used to observe living specimens of bacteria, although when using fixed specimens, bacteria have an optimum viewing magnification of 1000x. Bright field microscopy has very low contrast, and most cells absolutely have to be stained to be seen. Staining may introduce extraneous details into the specimen that should not be present. Also, the user will need to be knowledgeable in proper staining techniques. Lastly, this method requires a strong light source for high magnification applications, and intense lighting can produce heat that will damage specimens or kill living microorganisms. 2. Phase contrast light microscope. Phase contrast microscopy is a technique that makes invisible details in transparent samples visible. Frits Zernike, a Dutch physicist and mathematician, built the first phase contrast microscope in 1938. He won the Nobel Prize in 1953, and the German-based company Zeiss began manufacturing his phase contrast microscope during World War II. When light passes through a clear specimen, the light waves slow down or speed up depending on what they pass through. This changes the phase of the light, but not the brightness, so the human eye cannot see these changes. Normally, we only notice differences in brightness or color. Phase contrast microscopes take these hidden phase changes and turn them into differences in brightness. As a result, things that are normally invisible in clear, unstained cells suddenly appear as bright or dark areas, allowing us to see their structure without staining or killing the cells. Parts of the phase contrast microscope. The instrumentation of the phase contrast microscope is based on its light pathways from receiving the source of light to the visualization of the image. Therefore, it's sequentially made up of light source, mercury arc lamp, collective lens, aperture, condenser, condenser annular, specimen, objective, phase plate, deflected light, and phase ring. Working principle. The main idea behind phase contrast microscopy is to make tiny phase changes in light, changes we normally cannot see, visible as differences in brightness. To do this, the microscope separates two types of light. 1. Background light that passes through the specimen without being changed. 2. Scattered light that is affected by the specimen and contains the details we want to see. A ring-shaped beam of light is focused onto the specimen. Some of this light gets scattered by the specimen, but most passes straight through unchanged. In normal conditions, the scattered light is very weak and slightly out of phase compared to the background light, so both look almost equally bright. This gives a low contrast image. Phase contrast microscopes improve this by using two tricks. 1. Phase shifting. The background light is passed through a special ring, phase plate, that changes its phase by -90 degrees. This makes the background and scattered light line up better, increasing the brightness difference between them. 2. Dimming the background. A gray filter ring slightly reduces the background light's brightness so that the scattered light stands out more clearly. These two steps make the details of transparent, unstained cells much easier to see. This description explains negative phase contrast. In positive phase contrast, the background light is shifted by +90 degrees instead, making it 180 degrees out of phase with the scattered light. This causes the scattered light to reduce the background brightness, producing an image where the specimen appears darker than the background. Advantages: the capacity to observe living cells, and as such, the ability to examine cells in a natural state. Observing a living organism in its natural state or environment can provide far more information than specimens that need to be killed, fixed, or stained to view under a microscope. High contrast, high resolution images. Ideal for studying and interpreting thin specimens. Ability to combine with other means of observation, such as fluorescence. Modern phase contrast microscopes with CCD or CMOS computer devices can capture photo and video images. In addition, advances to the phase contrast microscope, especially those that incorporate technology, enable a scientist to hone in on minute internal structures of a particle and can even detect a mere small number of protein molecules. Disadvantages: annular rings limit the aperture to some extent, which decreases resolution. This method of observation is not ideal for thick organisms or particles. Thick specimens can appear distorted. Images may appear gray or green if white or green lights are used, respectively, resulting in poor photomicrography. Shade-off and halo effect, referred to a phase artifact. Shade-off occurs with larger particles, results in a steady reduction of contrast, moving from the center of the object toward its edges. Halo effect, where images are often surrounded by bright areas which obscure details along the perimeter of the specimen. Anodized phase contrast utilizes amplitude filters that contain neutral density films to minimize the halo effect. Essentially, this is attempting to reverse the definition achieved through phase contrast annuli, but the halo effect can never be eliminated completely. Applications of phase contrast microscope. Determine morphologies of living cells, such as plant and animal cells. Studying microbial motility and structures of locomotion to detect certain microbial elements, such as the bacterial endospores. 3. Dark field light microscope. This is a specialized type of bright field light microscope that has several similarities to the phase contrast microscope. To make a dark field microscope, place a dark field stop under the condenser lens. This setup changes the way light travels, so that the condenser sends a hollow cone of light shaped like a ring, toward the specimen. In dark field microscopy, only the light that is scattered by the specimen enters the objective lens. Light that does not hit the specimen does not reach the objective. This technique is used to visualize living unstained cells. This is affected by the way illumination is done on the specimen. In that, when a hollow cone beam of light is transmitted to the specimen, deviated light, unreflected or unrefracted, rays do not pass through the objectives, but the undeviated, reflected or refracted light passes through the objectives to the specimen, forming an image. This makes the surrounding field of the specimen appear black, while the specimen will appear illuminated. This is enabled by the dark background, thus the name dark field microscopy. Applications of the dark field microscope. It is used to visualize the internal organs of larger cells, such as the eukaryotic cells. Identification of bacterial cells with distinctive shapes, such as Treponema pallidum, a causative agent of syphilis. 4. Fluorescence light microscope. A fluorescence microscope is a type of optical microscope that creates images using fluorescence or phosphorescence, rather than just normal light reflection or absorption. In this method, certain parts of a sample give off light when they are excited by a specific wavelength. Any microscope that forms images using this fluorescent light is called a fluorescence microscope. This can include simple systems like an epifluorescence microscope, or more advanced ones like a confocal microscope, which uses special optical sectioning to produce sharper, high-resolution fluorescent images. Working principle. A fluorescence microscope works by shining a special kind of light onto a sample so that the sample glows. Certain dyes or molecules in the sample, called fluorophores, absorb the incoming light and then give off light of a different color. This glowing light is what the microscope sees and turns into an image. The microscope uses filters to make sure only the glowing light from the sample reaches your eyes or the camera. These filters block the strong light used for shining on the sample and let the weaker, colored glow pass through. A fluorescence microscope has a strong light source, such as a mercury lamp, xenon lamp, LED, or laser. The light passes through an excitation filter that chooses the exact color needed to make the sample glow. A dichroic mirror then directs this light onto the specimen. When the sample glows, the dichroic mirror allows this new colored light to pass through a different path. The emission filter ensures only the glowing light reaches the detector. Most fluorescence microscopes used today are epifluorescence microscopes, meaning the same lens both shines the light onto the sample and collects the glowing light. This design gives a clear, bright image because most of the extra light is filtered out, so only the glow from the sample is captured. If scientists want images with multiple colors, they take separate pictures using different filters and then combine them. This method is widely used in biology to study cells, tissues, and tiny structures that would normally be invisible. Light sources. Fluorescence microscopy requires intense, near-monochromatic illumination, which some widespread light sources, like halogen lamps, cannot provide. 4 main types of light source are used, including xenon arc lamps or mercury vapor lamps with an excitation filter, lasers, supercontinuum sources, and high-power LEDs. Lasers are most widely used for more complex fluorescence microscopy techniques, like confocal microscopy and total internal reflection fluorescence microscopy, while xenon lamps and mercury lamps and LEDs with a dichroic excitation filter are commonly used for wide-field epifluorescence microscopes. Sample preparation. A sample of herring sperm stained with SYBR green in a cuvette illuminated by blue light in an epifluorescence microscope. The SYBR green in the sample binds to the herring sperm DNA and, once bound, fluoresces, giving off green light when illuminated by blue light. In order for a sample to be suitable for fluorescence microscopy, it must be fluorescent. There are several methods of creating a fluorescent sample. The main techniques are labeling with fluorescent stains, or, in the case of biological samples, expression of a fluorescent protein. Alternatively, the intrinsic fluorescence of a sample, i.e. autofluorescence, can be used. In the life sciences, fluorescence microscopy is a powerful tool which allows the specific and sensitive staining of a specimen in order to detect the distribution of proteins or other molecules of interest. As a result, there is a diverse range of techniques for fluorescent staining of biological samples. Biological fluorescent stains. Many fluorescent stains have been designed for a range of biological molecules. Some of these are small molecules which are intrinsically fluorescent and bind a biological molecule of interest. Major examples of these are nucleic acid stains like DAPI and Hoechst, excited by UV wavelength light, and DRAC5 and DRAC7, optimally excited by red light, which all bind the minor groove of DNA, thus labeling the nuclei of cells. Others are drugs or toxins which bind specific cellular structures and have been derivatized with a fluorescent reporter. A major example of this class of fluorescent stain is phalloidin, which is used to stain actin fibers in mammalian cells. There are many fluorescent molecules called fluorophores or fluorochromes, such as fluorescein, alexafluors, or dilite 488, which can be chemically linked to a different molecule which binds the target of interest within the sample. Applications of the fluorescent microscope. Used in the visualization of bacterial agents such as Mycobacterium tuberculosis. Used to identify specific antibodies produced against bacterial antigens/pathogens in immunofluorescence techniques by labeling the antibodies with fluorochromes. Used in ecological studies to identify and observe microorganisms labeled by the fluorochromes. It can also be used to differentiate between dead and live bacteria by the color they emit when treated with special stains.